Well, good day to everyone joining us and welcome to today's XTalks webinar. Today's talk is entitled AI and Radiomics in Drug Development, Challenges and Approaches. My name is Samaya and it's my pleasure to be your XTalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Quibim, who developed the content for this presentation. At Quibim, they're revolutionizing personalized medicine by delving beyond what the human eye can see. They employ AI-powered imaging tools derived from years of intensive research to assist doctors in diagnosis and anticipating patient progression. Their solutions transform imaging data into actionable predictions, enhancing patient outcomes and driving medical imaging innovation. Now internationally expanded, Quibim is excited to introduce their new line of products focused on predictive imaging biomarker panels, ensuring continuous advancement in life sciences and provider solutions. Now it's my pleasure to introduce our speakers for today's event, and our first speaker is Dr. Ron Korn. Dr. Ron Korn, as founder and chief medical officer at Imaging Endpoints, has a Doctor of Philosophy degree in molecular biology at Albert Einstein College of Medicine, which provided a solid foundation for his medical training at Stanford School of Medicine, followed by postgraduate training at the Hospital of the University of Pennsylvania. In addition, medical director consultant in oncology at Virginia G. Piper Cancer Center at Honor Health, and adjunct professor at Translational Genomics City of Hope. Dr. Korn specializes in nuclear medicine and cancer biology with extensive involvement in clinical research as both a study investigator and clinical publication author that has afforded him in-depth knowledge of the common pitfalls and complex challenges associated with clinical trials. And our next speaker is Pamod Hemakumara. Pamod Hemakumara serves as the Director of Corporate Development at Quibim to drive inorganic growth, strategic partnerships, and fundraising activities at Quibim. His role also has a focus on growing Quibim's biopharmaceutical business, which involves partnering with biopharmaceutical companies for Quibim's novel radiomics and AI-based predictive algorithms. He holds degrees in biochemistry, biotechnology, as well as business administration. And our next speaker is Fuensanta Bellvís Bataller. Fuensanta Bellvís Bataller, as VP of Clinical Studies at Quibim, is responsible for the department that manages clinical trials, observational studies, and research projects in collaboration with biopharmaceutical companies and academic and research institutions, mainly in the areas of oncology, immunotherapy, rheumatology, and neurology. With an extensive technical background, she brings to clinical studies a scientific vision to optimize the exploitation of medical imaging and its transformation into actionable predictions. So now, without further ado, I'd like to pass control to our speaker. So Pamod, whenever you're ready, you can go ahead and get started.
Thank you, Samaya. Hopefully everyone can see my slides. So just a quick outline. In the initial section, I will go over some of the general trends and provide a very high-level overview of adoption of AI along the drug discovery and development process, both in terms of historic adoption of AI as well as future areas of interest. Then I will hand it off to Fuensanta to provide an introduction to Radiomics and AI, as well as cover the utility of it in clinical trials and in precision medicine. And finally, Dr. Ron Korn will go deeper into the use of radiomics and AI in clinical trials, especially from an oncology perspective. So before we go into the applications of AI, I want to quickly remind everyone of the standard drug development paradigm, or the discovery and development paradigm, which has several main stages. The first is the drug discovery and preclinical section, which involves identifying targets and drug candidate molecules, and then screening them to eventually test them in cell lines and animal models. Next is clinical development, which involves testing these medicines or future medicines in humans in a series of larger and larger trials that focus on establishing safety and efficacy parameters. And then you submit all of that data to the regulatory authorities to hopefully get approval to market the drug. And finally, there's a post-approval setting where you typically do surveillance and monitor the drug for any unforeseen or expected issues, including safety concerns. And the key thing here is this is a 10-year plus process that costs several $billion just to get a single drug approved. And this is important to keep in mind as we look at potential applications and use cases of AI, which can reduce these timelines, reduce these costs, and alleviate some of the struggles of drug development. So using this framework of the drug development process, it's quite clear that AI adoption has been quite profound over the last few decades in the drug discovery and early development stages. Because at these earlier stages, you're dealing with enormous amounts of data, potentially screening tens of thousands of compounds. And these type of processes are ripe for AI, and they can be streamlined and optimized using AI tools. And some examples at a very high level is in the target discovery and validation, which is one of the initial focus areas of the drug development process, where the goal is to find new disease targets to focus on as a potential drug target. And there are a plethora of data sets you can use to train your AI models, including gene expression, proteomic interactions, genomics, proteomics, as well as patient registry data, which can be systematically mined using AI algorithms to hopefully identify novel disease pathways and targets. Then the second area of AI adoption that I want to cover in this early stage is the hit and lead identification and optimization. The goal here is to generate molecules and predict if these molecules can interact with targets based on various properties like structural and biochemical properties. So at this point, you have the disease target identified, and it's a matter of finding molecules that can interact with the target or bind to the target in a particular way that hopefully elicits the response that you're looking for. The type of data sets you can rely on for these type of AI algorithms may include chemical, biochemical, and physiochemical data sets. So in addition to this, in theory, you can also use AI approaches to predict various characteristics of a molecule, including its toxicity, pharmacodynamics, pharmacokinetics, and other aspects that are important in the later stages of the development process. So essentially, you're trying to screen the molecules early on before they get tested in more expensive and time-consuming processes like cell lines and later animal models, and eventually in humans. So you can, in theory, also use AI approaches to predict the safety and efficacy in humans, although this is not a use case that's been developed and adopted to a high degree just yet, because it's very difficult to do. However, in theory, you do have access to all the preclinical data for the molecule of interest, as well as potentially a variety of preclinical and clinical data on similar compounds, which you can use to develop algorithms using AI. Just to provide a few examples from the real world, we have, in the case of target discovery and validation, which is actually also an example of drug repurposing. We have the company Benevolent AI that was able to take advantage of the COVID-19 pandemic to mine a set of structured data sets of machine-read literature to discover an existing drug that had antiviral properties. This drug was baricitinib, which is an approved JAK inhibitor for rheumatoid arthritis. And they found that this drug, in addition to its anti-inflammatory effects through the JAK pathway, it had antiviral properties by mediating the entry of the COVID-19 virus into cells. And this was apparently done in a matter of days. So that's very powerful when you think about how long drug development takes for this stage in a typical context. Another example is the company Exscientia, which was able to fully design a molecule for a known target. So this is an example of hit and lead identification optimization, in that they were able to take an existing drug target that was well understood and design a molecule that would interact in a way with that target to elicit a response that is favorable. And in this case, it was a serotonin 5-HT1A receptor agonist, meaning the AI developed compound binds to the receptor and also changes the receptor's activity to elicit a response. And this, I believe, was the first example of a fully AI designed compound that entered clinical trials. Unfortunately, it didn't make it past phase 1 as it was discontinued, but since then, there have been many cases of AI developed or designed molecules that have entered the clinic. And of course, this market, given the use cases and the real world applications of AI and drug discovery, is a multi-billion-dollar market. It's growing quite fast at least a 25% annual growth rate. Oncology has been the primary therapeutic area of focus, but it's important to note that infectious disease is expected to grow quite rapidly in the future. And it's also important to keep in mind that, as is with most AI applications, the access to suitable data sets of high enough quality and quantity will remain one of the limiting factors in the growth of the space. And of course, it's still evolving and standardizing, and there could be new use cases or existing use cases that will see quick advancements, which will ultimately contribute to the growth of the space. So it's important to stay up to date on all the advancements if you want to know where this is all headed. So moving on to the clinical development, regulatory review, and post-approval settings, there are some emerging concepts that have led to interesting AI applications. And the one I want to talk about is real-world data and real-world evidence, which has been a growing topic, especially from a regulatory perspective, because insights generated from real-world evidence and real-world data can help make informed decisions in various aspects of drug development and commercialization, including regulatory submissions and post-surveillance settings. So real-world evidence can be defined as quote-unquote clinical evidence regarding the use and potential benefits and risks of a medical product derived from real-world data. So what is real-world data? It can come from data sets such as electronic health records, administrative billing records, and insurance claims, and any real-world data in an essence, right? And the insights generated from this data can provide insights into aspects such as adverse events detection, outcomes research, comparative effectiveness, and safety surveillance. And real-world evidence has been used to confirm and support regulatory submissions for many top therapeutics. And again, the availability, the usability, and the shareability of high-quality data will remain a major challenge. And one of the AI, or in this case, machine learning technologies that have greatly enabled real-world evidence generation is natural language processing, or NLP, which essentially allows machines to understand and communicate in natural language. So for example, in text, audio, images, things of that nature. This has enabled the creation of structured data sets that are based on large quantity of electronic health record and billing info that otherwise would be very tedious and quite frankly expensive to do if it were done by humans. And the real-world evidence generated from NLP has led to several use cases spanning, as I mentioned earlier, adverse event detection, optimization of recruitment in clinical trials, and drug repurposing. And additionally, in post-approval settings, there have been several AI applications across pharmacovigilance, predictive analytics, quality control, and compliance. And there are some examples here on the right-hand side, which I will not go into. And real-world evidence as an industry has emerged quite strongly in the last decade or so with companies like Flatiron Health, for example, that have made a good business out of creating structured data sets. So now let's cover some of the emerging technologies that apply to clinical and post-approval / commercial stages. And this will be kind of a high-level background, and Fuensanta will go into detail into some of this later on, as well as Dr. Korn. So there are many different focus areas for optimizing clinical development, right? It can range from integrating digital health technologies to using novel biomarkers, optimizing trial design, and several other aspects. One technology that shows promise in this space is radiomics and AI-based algorithms that can be used as a prognostic and predictive tool. So the idea here is you design an algorithm that gives you a desired prediction, but the input of getting that prediction is simply a medical image in this case, and potentially a few other parameters. But the idea is you take a baseline scan and you gain insights based on that beyond what you can generate today. So these have various use cases, which include guiding clinical treatment decision-making, improving clinical trial design, and optimizing drug development programs. In practice, these algorithms can act as two types of products, very broadly speaking. First is drug development tools, which can be used in clinical development. And the second is companion diagnostics or digital companion diagnostics. Similar to how there are genetic-based companion diagnostics through companies like Foundation Medicine, we believe that there's a tremendous opportunity for imaging-based digital companion diagnostics. And ultimately, these radiomic and AI-based approaches can enable precision medicine at a much wider scale. So before we go deeper into radiomics and AI-based approaches, I want to quickly summarize this first section in that, we established drug development is costly, it's time-consuming, it's very inefficient if you look at it in a way of success rates, which is ripe for AI adoption. And this AI adoption has initially happened at the drug discovery and the early development stages, which is a multi-billion-dollar market that's growing rapidly. But there's this large window of opportunities for new applications of AI, as well as the existing applications to, advance further and improve. And one of the ones that show a lot of promise, of course, is radiomics and AI-based companion diagnostics and drug development tools, which Fuen will go into in the next section. And the final thing to keep in mind is that data quality, quantity, and shareability, as well as accessibility, will remain important levers in the adoption rate of AI. So with that, I'll hand it off to Fuen. Thank you.
Okay, thank you so much, Sumaya and Pamod. Hopefully all is going well and you are seeing my screen in full mode. So after listening what my colleague Pamod was mentioning about the radiomics and AI-based technologies used as DDTs or CDX that represent this large opportunity to optimize the overall drug development programs, I would like to go further to the fundamentals. That's why we will start my presentation explaining to you what is radiomics and which is the difference between radiomics and imaging biomarkers, how the couple of radiomics and AI can add a lot of value to this drug development and clinical trials scenarios, and how these technologies are able to advance precision medicine and to have a pivotal role in clinical trials, being a new protagonist in the jungle of response criteria. I will try to explain a case study, mainly focused on immune checkpoint inhibitors, and we will finalize with the current challenges and future prospects that Dr. Korn will take to go further. So radiomics is a cutting-edge field that allows the extraction of high number of quantitative information from different kinds of medical images, ranging from intensity distribution and spatial heterogeneity to higher order statistical parameters. In the opposite side, an imaging biomarker is as well a measurable sign, but in that case, it's linked and related to a biological state or condition. So in the right side of the slide, you can see an example of a radiomic fetus. We have a patient with advanced non-small cell lung cancer, in which we have detected the lesion using an AI tool. Apparently, this lesion seems to be an homogeneous one, but once we extract this quantitative information that cannot be perceived by the human eye, we can see that this tissue is not as homogeneous as we thought. And here you can see this parametric map showing different levels of, in that case, entropy in the lesion. In the second row, you can see an example of an imaging biomarker. For example, the ADC apparent diffusion coefficient is extracted from the diffusion-weighted images and is linked to the cellularity of the tissue. Or the proton density fat fraction, which is extracted from another kind of MRI, from the MEXA sequences, and allows us to quantify the fat that is distributed in the overall liver. Or the standardized uptake value that we can extract from the PET images, which is linked to the metabolism of the body. So all these techniques, radiomics and imaging biomarkers, allow us to convert images into mineable data that advanced statistics or AI, as you will see later, can be applied to generate actionable insights and to have actionable predictions. So according to the FDA NIH Biomarker Working Group, there are 7 essential categories for the biomarkers. But here you will see that we have focused on 6 because these are the ones that are highly related to the imaging biomarkers. So we can use this quantitative information, for example, to develop a model that allows to predict the risk of suffering a disease or the risk of having the disease to suffer metastases. Or we can use these techniques to detect a disease in an early scenario or in an efficient way, or to identify individuals that has a subtype of the disease that can be linked to a specific treatment approach. We can also talk about prognosticability in the sense that we can predict a clinical event. We can predict overall survival, progression-free survival, among others. Or we can go further to the treatment. And in that case, we can talk about predictive biomarkers. This allows to predict which is going to be the response of these patients, or if they are experience a favorable or unfavorable effect after being exposed to a specific dose. But that's not all, because at the end, once a drug is given to the patient, several biological processes are occurring on the body, and we can also link these biological responses to a specific imaging biomarkers, coming back to what my colleague Pamod was mentioning about pharmacodynamic biomarkers or surrogate endpoint biomarkers. And last but not least, we can also talk about the safety biomarkers in the sense that we can use this quantitative information to know the likelihood of suffering an adverse effect. For example, suffering a neurotoxicity in a specific treatment like CAR T therapy, among other examples that we can share. But what occurs with radiomics if it's not coupled with AI? At the end, artificial intelligence is giving us the chance to go further and not only extract quantitative information using radiomics and imaging biomarkers, but also using these techniques, mainly the CNNs, to extract the deep features that can be combined. We can extract thousands of deep features that can be combined with the radiomics and imaging biomarkers to have a further explanation of what is occurring in the body once we see within the tumor, but also in the tumor microenvironment, that as important as we know it is. And on the other hand, AI can allow us to handle the massive amount of data, because at the end, if we only use the radiomics information, we are not processing them. So we need to use advanced statistics, or if we want to go further and handle and deal with a massive amount of data, we can use AI to combine this information and to be able to apply this radiomics to predictability. So we can use the combination to extract and analyze this meaningful hidden quantitative information that can be used as CDSS or our clinical decision support system, and then to allow us to use this radiomics and quantitative information. But can radiomics and AI enable precision medicine? The genomics and pathomics have shown during the last year significant breakthroughs in precision medicine. They have been able to stratify patients in different subpopulations. They increase safety and efficacy of targeted therapies, and have shown an increased efficiency of clinical trials. In fact, 34% of the new drug approvals in 2022 were done thanks to this genomics and pathomics applicability. But there are some limitations. For example, there is a minimum number of cells that are required once we have a sample of a tissue. And there is also a bias, which depends on the location of the needle once we perform the biopsy. And biopsies are performed only once. And apart from that, there are logistical limitations that are reducing patient access. For example, here you can see a figure of 64% of eligible advanced non-small cell lung cancer patients are not benefiting from precision oncology treatments due to not being able to have access to this next generation sequencing and further techniques that allows to process and extract this genomic and pathomic information. In this regards, the imaging-based solutions are offered as a complementary information. At the end, imaging is a standard of care procedure that can be universally accessible and performed at the multiple steps of the disease. So having these images, we can be able to capture heterogeneity within and beyond tumor, which is an important limitation of the molecular techniques. And this is non-invasive in the sense of not being able to perform a needle, for example, on an organ of the patient or on the lesion of the patient. This is measured at every step of the patient journey. So it's provided as an scalable, accessible, and affordable solution that can be complemented with pathomics and genomics, increasing the overall patient access to precision medicine. But let's move on in the advantages of radiomics analysis once they are applied into the clinical trials scenarios. First of all, with these techniques, and when I talk about radiomics, I'm also considering the combination of radiomics and AI, we can be able to exploit the data that is being acquired in the clinical studies. So we can discover more about medical images that not always are fully exploited, and we can make it through a non-invasive and cost-effective tool. And this can be applied not only in oncological conditions, but also in other kinds of neurological disorders or chronic diseases. On the second step, we can also unleash the insights about the characteristics and progress of the lesions and tumors going beyond the traditional reading methods. So with this kind of techniques, we can not only evaluate the tumor, the lesion, or the organ in a 2D measurement, we can evaluate the overall tissue, and not only the tumoral tissue, but also the microenvironment that goes behind this. And in the third position, we can evaluate the lesion tracking. We can know how the lesion is changing longitudinally, which allows to perform a monitoring and a clear trajectory of the disease progression or the response to the treatment. So now that we have already been discussing about what is radiomics, why it's important to combine radiomics with AI, and how radiomics is being able to exploit and quantify data that is covered or inside the medical images, I would like to explain you real examples of how the combination of these techniques can accelerate the drug development programs. And I will try to do it starting from the beginning or from the starting point of a clinical trial, ending up with the post-market strategy and the final phases of these studies. So advanced radiomics can allow a better trial recruitment. So we can use this information extracted from the images to evaluate beforehand to which treatment arm the patient needs to go. So we can know further and we can evaluate from the beginning of the clinical trial design which is the best assignation for these patients or for these subjects. Innovation in early development. We can also use the radiomic features to boost value and investment potential. So we can evaluate the success rate of the clinical trial in the early development and to take decisions having quantitative and measurable information. Detailed evaluation of toxicity and dosimetry. Radiomics can be also used to evaluate which is the maximum tolerated dose, and also in escalation dose studies, this can be used to know which is the specific optimum dose for the patient, and then to evaluate which needs to be the second step for further phases. Targeted patient stratification. Using radiomics, we can accurately predict which patients are going to benefit best from a specific treatment. And this can allow us to know, for example, if a specific subpopulation of a specific treatment target is going to suffer an early progression. Are there critical evaluations? Once we are advancing and we are talking about phases 2 and 3, Radiomics and AI can also bring further information to have a comprehensive safety and efficacy analysis. So the radiomics can be used to evaluate which is the optimum point of moving forward to the second step of the clinical trial. And the data harmonization for reproducible outcomes. So mainly in advanced clinical trials, I'm mainly referring to phase 2, phase 3 clinical trials in which we are handling data that belongs to different manufacturers with different imaging acquisition protocols, field strengths among the likes. It's very important to apply standardizing imaging quality techniques that allows to remove this bias and to focus on the quantitative information that depends on the biological processes and not onto the scanner effects that can be linked to the specific acquisition process. So we can also talk about end-to-end AI-based radiomic solutions. So these radiomic solutions can be used as exploratory endpoints in a clinical trial. And this can give information offering a more straightforward path to regulatory approvals and market entry. And finalizing, we can expedite regulatory approval. So having all this information in a robust way and in a solid manner can allow to meet the demands of the regulatory authorities in a most speed up way and having all the information in place before starting the FDA approvals and processes. So let me share now an example of all this. Now we have been talking about several applications, but I would like to share a success use case that we have been working at the company, which is mainly focused on the prediction of response to immunotherapy using, on that case, baseline CT scans. So we are talking about advanced non-small cell lung cancer patients. In that case, it's a phase 3 trial in which we have, as you can see, thousands of CT scans and different sites and scanner models. So here you can guess that the harmonization in this regard was very, very powerful, very important. And at the end, immune checkpoint inhibitors have demonstrated better results in terms of treating patients with non-small cell lung cancer. But there is an unmet need to identify an ideal biomarker for predicting its response. So in this regard, the solution that we developed together with the biopharmaceutical company was using radiomics that are extracted from the baseline CT scans to allow the prediction of this immunocheckpoint inhibitor response. So this allows to go beyond the traditional measurements that, as I was mentioning before, have some limitations, and also to evaluate in a non-invasive manner the tumor microenvironment, its heterogeneity, moving forward with the prediction of the overall survival. So here you can see an example of this clinical study in which apparently the lesion seems homogeneous, but once we extract the quantitative information, and in that case, this is a specific radiomic feature, and we represented it, we can see that this is not as homogeneous as it seems. So all the information that was extracted in this, from these CT scans, which belongs to radiomic features, to imaging biomarkers, to deep features, were combined to create predictive models that allows to know the best overall response of the patient, overall survival, progression-free survival, and the early progression. So we have been using radiomics information combined with AI to be able to predict at the patient level and also at the lesion level how the treatment is affecting the subject. But as you can imagine, there are still challenges that needs to be addressed. And here I would like to share some suggestions and future prospects trying to address them. So first of all, the majority of publications related to radiomics and AI are using a small cohorts from single centers. This is putting us now the need of creating multi-center real-world evidence studies. So we need to have further analysis that has a higher sample size and that allows to create more generalizability models. In addition, there is the heterogeneity of the data. Now, these scanner effects I was mentioning before, it's very important to harmonize the imaging and the features information to be able to remove the bias that is linked to the scanning or the acquisition parameters, and to make sure that all the quantitative information that is being extracted and processed is linked to a specific predictability and a specific biological process. Repeatability and reproducibility, this is another important aspect. We need to detail all the workflows and perform solid developments to make sure that once we perform or make inference of these models, we will provide or we will extract the same information. Result sharing, it's important to not only share positive results, but also results that are negative and that have bring up the possibility to know that this is not the right way to go. Or interpretability, all of us have been listening about the black box, so it's very important that our models goes hand by hand with an interpretability and explainability techniques that allows to know what is occurring, what is the meaning of a specific deep feature or a specific radiomic features applied to this aggressiveness of the tumor. Or the validation. At the end, it's important to validate using external cohorts all the algorithms that are being developed. And that's ideal in real-world evidence studies. And finally, the accessibility. It's very important to be able to access databases that belongs to different institutions and to have a big anonymized data lake that allows this further validation and at the end to put these radiomics tools in clinical adoption and applicability. So I would like to share final key takeaways from my presentation. So radiomics and AI allow the extraction and analysis of meaningful quantitative imaging data, offering new insights for lesions characterizations, tracking, and progression. As you have seen, radiomics can open a new door to life sciences companies exploiting the imaging in their pipelines, and is expected to play a pivotal role in enabling precision medicine. Radiomics and AI can optimize drug development programs using all these screening and stratification options, evaluating toxicity and dosimetry, and offering a more straightforward path to regulatory approvals and market entry. And the development of end-to-end AI-based radiomic solutions can allow to identify biomarkers for predicted treatment response and advanced patient outcomes, although some additional challenges need to be addressed. So thank you so much, and looking forward to the questions. So I will handle it to Dr. Korn.
Thank you very much, Fuen, for a great presentation. I am trying to share my screen here, and hopefully you can now see my screen. Well, I really appreciate Pamod's and Fuen's explanations of the field of radiomics and AI. I think they were exceptional explanations. And so for the next 15 minutes or so, I'm going to review 3 areas that I'll comment a little bit further on. First is, I think Fuen did a great job in reviewing the challenges of integrating AI and radiomics in clinical trials. I'll just highlight a few more points from what she presented. Then I want to show you 2 use case examples that we've used here at Imaging Endpoints to really show the value of radiomics and AI in clinical trials. And then finally, discuss some opportunities that I think are almost right there, or if not already being applied to the use of AI and radiomics in clinical trials. And I like to put the context of how we use radiomics and AI in our imaging core lab. The way that we use it is that we provide very comprehensive ICRO services, and most of our investigators, most of our sponsors, most of the biotech and pharmaceutical companies are now asking us to integrate these advanced imaging analysis, such as radiomics and AI, into our blinded independent central review response efficacy workflows. And that's a really important gain of efficiency, as you can well imagine, as you're doing the required RECIST reads or Lugano reads or Ronald reads, depending on the tumor type. You really want to also identify those same lesions that you're following on these examinations for radiomics. And so we do have an all-in-one way of doing that. And in this process, we have received one of the highest marketing authorization rates in the imaging CRO space as we advance these different types of imaging analysis towards registration. And we work very collaboratively with our partners at Quibim, especially when we do specific customized and advanced image analysis. So Fuen highlighted many of these challenges that we have in radiomics. I want to just touch upon a few of these. The first is evolving technologies, and this especially occurs in the arena of PET imaging, where now PET radiomics is becoming a very important aspect to understand. As you know, PET scans are changing their technologies. It started out with the standard technologies with a single bed position. Now the new scanners are digital scanners, which do whole body imaging at one time. And so the signals are quite different than what they are in the standards. So when you set up a retrospective review of looking at standard PET radiomics, you have to then account for the new evolving technologies. And so that's not always straightforward or easily accomplished. As you know, there are a variety of different ways of doing radiomics, different platforms, Pi Radiomics, Quibim Radiomics, and each of these platforms really sometimes provide different sorts of imaging features or extraction of data. So how do you handle that? And for here, we really do look at the imaging biomarker standardization initiatives, in which you try to find, well, what's the common way of analyzing these radiomic images across platforms so you can compare them. One of the most important things that I find in getting not only our clinicians, but clinical sponsors to really adopt radiomics in full-fledged is they understand the mathematics, they understand how these can be evaluated with AI and statistics, but what's driving the biology? Because fundamentally, that is going to really make radiomics successful if we really understand the biology. And for this, we've been doing several preclinical studies. Members of my scientific group have been doing preclinical studies and then translating them into the clinical trial studies. That, I think, is one of the areas that needs to be solved here in the next several years. Fuen talked about the black box and non-transparency. I had nothing more to add. And the validation, just to keep in mind that there are two validations we talk about. There's the scientific validation, which we're all working very hard in which to prove that these are effective means, robust means of evaluating changes on imaging. But there's also the regulatory environment. And the regulatory environment asks for accuracy. If we're going to use them, how accurate are they in making decisions and making predictions? So that is one of the key aspects of regulatory evaluation. And then, of course, being able to scale and make it radiomics universal is very important. Well, here at Imaging Endpoints, the way that we integrate radiomics in clinical trials really shown here on this slide. We consider 6 steps here. The very first step, of course, is what question are you asking? What is your goal? What is your objectives of doing radiomics? Once you understand that, then it's essential in any clinical trial that you have the correct documents in place that will allow you to do the analysis in a prospective manner as the best practices. So that means that you have to make sure your clinical study design is correct, that your site imaging manuals that you provide to the site, even if it's a single center or multi-center, is set up for allowing the standardization of imaging acquisition. How you're going to evaluate those images to the charter is really critical to the success. What are you going to do with the data? What's your analysis plan? And we always do recommend that even though it's not necessary when you deal with de-identified images to make sure that the patients have it in their conformed consent, we think that it's best practices to make sure that there's some language in your documents, that informed consent, that allows Quibim and ourselves to do the radiomics. Once that's complete, of course, site preparation. It's important to understand, well, what equipment is going to be used, what sites are going to be involved in the trial. Real world data is critical, but I think when you're in a clinical trial, these are essential things to understand. We believe a lot in phantom harmonization to make sure that whatever scanners you use are performing about equally across all centers. And I think that the site personnel understanding what we're trying to accomplish there really helps to get a better product at the end of the day. Once you get into study conduct, of course, you have to make sure that the patients are and all the patient-centric issues are well considered, especially if you're going to be using PET, to make sure that you're getting the images at the same time during the following the injection of radioisotope, for example. Again, the technologies need to be harmonized. How are you going to process and acquire and process those images? How are you going to store them? And how are you going to do the data gathering? Because radiomics in isolation of the other information is not as powerful as when you can get all the data together. And there's some restrictions at sites that you work with that although the images have to be de-identified, somehow they have to be tagged or marked so they can be linked to the clinical data later. Fuen discussed the imaging analysis part. I won't go over how you build a radiomic signature. And then finally, reporting. Reporting is very important to not only the agency, but if you want to do publications in the standards that you must maintain. So in the last few minutes, let me give you a couple use case examples of where we at Imaging Endpoints have integrated radiomics into our blinded independent central review to show you how helpful this could be. So in this case, the sponsor wanted to use radiomics to help determine optimum dose during a phase 1 trial. As you know, the agencies are not pushing for maximum tolerated dose only. Now they want to know what is the maximum effective dose in early phase trials instead of going for the MTD alone. So in this case, we had about 160 patients that were undergoing blinded independent resist reads. And we, when we do radiomics here, we integrate it with tumor growth kinetics, which is an exponential which we use an exponential evaluation to look at whether there's tumor growth or decay based on the work of Tito Foyo at Columbia University. And here we looked at the last 3 cohorts of the dose escalation trial. We analyzed the whole tumor and used the first and second order radiomic features at baseline and first follow-up. We then constructed the model. And what we found that for response, that there were 5 radiomic features that made up the strongest model. And that when we combine that with our doubling time based on tumor growth kinetics, as you can see here, the longer the doubling time, the better. And dose level 2 was best, not only from the tumor growth kinetic part because it prolonged doubling time, but also when we look at the strength of the radiomic feature changes within that second dose level, it was strongest at that dose level. So this was really important information that showed that dose level 2 was more effective than dose level 3 and had a lower toxicity ultimately. Now the second use case is shown here. This one was really very interesting to me. And this was a maintenance trial for GI malignancies in which we were looking at event-free survival. So you get the cancer to its deepest response possible, and then the patient goes on a maintenance therapy to prevent any further deterioration in the tumor burden. And so in this trial, it was an open-labeled and importantly a randomized 1-to-1 comparing drug X to standard of care to prevent tumor progression. We looked at radiomics, both at baseline and the first follow-up, looking at whole tumor volume segmentation. And in this case, we looked at liver because this drug was metabolized through the liver. And so we thought perhaps the liver would also be important to understand the maintenance of the tumor burden. We used the radiomics models as shown here, and the results were really interesting. Although there was a modest difference between drug and standard of care favoring the drug, it wasn't statistically significant, though it was trending in that way. But what's really interesting, and shown here in the confusion matrix and also in the SHAPS diagram here, is that without knowing anything, just looking at the baseline radiomic data of either of both the tumor and the liver, we were able to determine, without knowing anything, what kind of treatment those participants were going to be subjected to. And so it meant that the radiomics was showing that there was a bias in the randomization, or a potential bias in the randomization process, even though the randomization is what you would consider a standard or classic randomization for any clinical trial. So what it told us is, and even though it's a small data set here, what it said is that radiomics can really be very helpful in many different ways that are yet to be explored, such as ensuring that if you have a randomization of a clinical trial, that you can think about using radiomics to ensure that you have a balance of the radiomic features of tumors between the two trials, so you don't get into this biased type of situation. And that ultimately leads to better subject stratification. So in conclusion, just to review now, what are some of the potential opportunities? What does the future look like? And this is how we're using the future of AI and radiomics in our core lab. First of all, when we get involved in evaluating your clinical study protocols for the imaging sections of your trial, we do use AI and some natural language APIs to look at what is out there in the literature compared to what you're going to be doing, to see if there are any differences or any things that we should consider that we haven't considered, especially in trials that have been conducted successfully. So AI is very useful to going out to the broader world and getting that information. We find AI is really helpful because we are in hundreds of sites around the world, thousands of sites around the world in hundreds of countries, and every site has a certain characteristic or personality, if you will. And so AI really helps us to understand, well, which sites are better in readiness or doing their site evaluation than others. And how do they perform? How many queries do we get in site X versus all the other sites? And that really helps to make sure the images are obtained more consistently. One of the things that we're really interested in, it hasn't come true, but I think in the next 5 years, we're going to see how we can use AI to turn a non-contrast image to a contrast-enhanced image. I am really convinced that this is one of the areas of great opportunity for AI. Imagine if you get a clinical trial and a quarter of your patients don't have contrast of the CT and you're being asked to evaluate that for RECIST. Wouldn't it be nice to see what it would look like if it was contrast-enhanced? We use AI a lot to understand the image QC process when they come in, to see what are some of the things in the DICOM tags that really need to be evaluated and addressed to make sure there's good standardization. We use it also to understand our radiologists' performance rates. Of course, I think that is alluded to by Fuen. If we have real-world data and we can analyze those by radiomics, now we really have a control in which to compare our study drug to the real world. And those synthetic controls are going to be driven by AI and radiomics. Of course, people always say that AI is going to replace a radiologist. Well, okay, I'll take a bit of a bet against that. In some cases it does, such as looking at normal chest x-rays versus abnormal, especially if a big list to look through. I mean, AI is great for that. But for lesion detection, it's a little more complex. I think PET is one of the areas in which it will be most useful. And of course, when you do your data management, your data control, looking for consistencies and inconsistencies, AI would really be very helpful. So in conclusion, I think we all are here because we understand that radiomics is a very powerful technique. It's going to really drive future discoveries. It's going to improve trial efficacy and approval rates. I think all recognize that there are challenges that still have to be met and overcome, and that advances in the technology of AI and machine learning will really get us there. I haven't touched upon the legal and the financial and the educational challenges that exist with AI. They are definitely out there and they need to be addressed. And I think the easiest one really is the educational challenges. Many people still don't know what radiomics is, what AI is. They're frightened, or they're very frightened about it. And this is where we need to have better education of what it can and cannot do. We believe here at Imaging Endpoint that a well-disciplined strategic approach is the way to go when you have to integrate AI and radiomics. And of course, we're here to help accelerate these advances as we connect imaging to the cure. And with that, I thank you very much for your attention and for questions. Thank you.
Well, thank you very much for that insightful presentation. And yes, that does bring us to a Q&A portion of this webinar. So attendees, if you have questions, keep sending them in. So we've already received some questions. So I'll start off with that first one. So Fuensanta, this one is for you. And that question is, how could a pharmaceutical company make use of the potential of radiomics and AI applied to medical images of a specific clinical trial, i.e., how could this work in a real setting?
Thank you very much for the question. So here, I would like to talk about the platform at the end, no? Because we have been talking about the applicability of radiomics and AI, but it's important to have a framework in which we have to start using a medical imaging that has been just a kid in a site, and then to be able to have the response that can be the overall survival or that can be the treatment response straightforward. So in this regard, it's very important that all these end-to-end AI solutions that can be created are integrated in a platform. And this can allow to closing the loop and starting from an imaging, ending up with a prediction of a patient level. So I think it's very important to have in mind and take into consideration that all these techniques needs to be embedded in a software that at the end allows its use.
Thank you. And another interesting question from an audience member. That question is for you, Pamod. That question is, what is your opinion on combining radiomics with other omics and clinical, molecular, etc. variables available to the patient?
Yeah, thank you. So my opinion is it is an interesting and most likely a valuable exercise to combine different data sets and modalities the goal here would be to improve the predictability or the performance of an algorithm, right? So and then that's something Quibim also focuses on, right? We don't want to just use the imaging data alone. Of course, that has a lot of value and shows a lot of promise, but you want to correlate that with other data sets that is more grounded to the foundations of medicine. So that could be genomics, could be pathomics, and other omics. All can be combined, and I think the ultimate goal, like I said, is to improve the predictability and performance of the algorithms.
Thank you, Pamod. And we have another great question from an audience member, and that's for you, Fuensanta. And that question is, regarding the data harmonization, in which phase of clinical trials would it be most relevant? Has it been applied in any collaboration Quibim has?
Okay, thank you again for that question. So mainly I have been sharing it during my pitch, but I think it's very important once we are talking about multicentric studies. So once we are including different sites with different scanners and different imaging acquisition protocols, in that moment, image harmonization and features harmonization takes an important role. And also we have been using it in some phase 3 clinical trials, and we have been able to compare, for example, radiomic features, like can be the entropy of the tissue, and just to see if the manufacturer is affecting or it's introducing a bias. The response was yes, that we saw that there is an important effect of this different manufacturer on the radiomic features. And then we compare what occurs once we use the harmonized data set versus the non-harmonized data set. And we concluded that these predictive models were more robust and more generalizable once we use the harmonized radiomic features. So I think that's an important step that we cannot avoid in that kind of studies, mainly once we are talking about phase 2 or phase 3 clinical trials.
Thank you very much. And a question for you, Dr. Korn. So that question is, how close are we for radiomics being accepted as a primary endpoint in a pivotal trial?
Great. Well, thank you for that question. We are close, but not close enough for all of this in the radiomic field. The agencies, the regulatory agencies, really require three things to occur, because at the end of the day, remember, they're there to evaluate safety, efficacy, and actionability. And there are three things that really need to be met before it will be accepted as a primary endpoint. The first is that we do have to understand the biologic meaning of the radiomic signature. That, I think, is very crucial to an understanding of what is it that we're measuring in these scans. What's going on at the cellular level, for example. And that, I think, is something that we're getting closer to understand, although those trials are still in their infancy. The second is that we really need a well-designed randomized controlled trial that really shows that radiomics as a primary endpoint predicts outcomes. And outcomes is not response. Remember, response is just a surrogate that the patients are going to do better. But they really want to see it in terms of looking at progression-free survival, event-free survival, and overall survival. And some of the data looks pretty interesting, that radiomics is a better predictor of overall survival than, for example, tumor shrinkage on RECIST. But it has to be in a well-designed, randomized controlled trial. And I think that's going to require a collaboration between industry, academia, and the private sector like ourselves. And the last thing that really accelerates approval is getting radiomics on a guideline, an NCCN guideline. If you can get it on an NCCN guideline, it really drives approval as using as a primary endpoint. We've seen this in other examples, and I think this will be true with radiomics. So I'm hoping that we're a half decade away from that. Probably will be plus or minus a couple of years.
Well, thank you very much for all of those answers. We've reached the end of the Q&A portion of this webinar. Attendees, if you do have more questions and comments, you can use the contact information on your screens. So go ahead and quickly jot this down. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Additionally, there's a link to view the recording of this event in the chat box. There you can, of course, share with your colleagues. The recording takes about one business day to 3 business days for that to be uploaded. So be sure to take a look out for that. Now, please join us in giving a big thank you to our speakers today. So that's Pamod, Hemakumara, Fuensanta, Bellvís, Bataller, and Dr. Ron Korn for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.
Thank you.
Thank you.
Thank you, everybody.